Welcome everybody. This video is for the American Association of Physics Teachers winter meeting in January 2021. And the topic is some materials that I've been developing for a textbook which is going to be called Biophysics and Physiological Modeling. But the focus of the talk is going to be for Introductory Physics for Life Science majors. And the thesis of the talk is that physics is the kind of physics that life science students want and need. So there have been a number of reports over the years focusing on why new materials might be needed. The first one focused on what students would need to be prepared for biomedical research. The second one was about pre-meds. The third one was about changing the whole biology major to focus on new requirements. This fourth one was a report for President Obama about preparing students for STEM in general. And the last one is the report from a working conference from the American Association of Physics Teachers about the Introductory Physics for Life Sciences course. An overarching theme was that students need to be better prepared in quantitative skills and even in computing. The AAPT IPLS recommendations suggested that we should include more stochastic processes such as diffusion, osmosis, radioactive decay, and the major challenge was to make thermodynamics more relevant to life science students. In order to add things to the curriculum, you needed to be able to remove things, and these are a few of the topics that were suggested that could be removed. Okay, so one of the issues with the traditional physics course is that it starts out with topics that most life science students don't find very interesting. They're kind of boring. And it takes until, week 4 or 5 before you get to things that apply to biomechanics, which is something that they might actually be interested in. Also, there's a problem with these initial topics because education research has shown that there are many student misconceptions about how motion really occurs, and starting out with that very mathematical concept right at the beginning, I think creates a problem that's very difficult to overcome. The second semester of the introductory physics course also starts with topics that students kind of don't really see why, light bulbs and batteries, why are those important for life science majors? So here's a list of topics that I think are of interest to life science students. We've already talked about diffusion and osmosis. Clearly, uptake of oxygen is important to aerobic organisms. Keeping cool is something that humans do by perspiring, and evaporative cooling is important. Drug elimination is important for pharmacy students and for any health science students. Radioactive decay, which we've already talked about. The founding principle of biochemistry and molecular biology is about ligand binding, and neuroscience and electrophysiology are important for the nervous system and how nerve impulses travel through the body. Biomechanics is a topic that fits fairly cleanly into traditional physics, and so I'm not going to talk about that here. And then finally, current events, students might be interested in COVID-19 and how to model it using epidemiological models. Topics that students need include quantitative and computing skills that they can easily get with Excel, which has the advantage of being a platform that they're not scared of. In order to be able to communicate with each other, talking about algorithms is actually really important. Another important concept that life science students need is to be able to model change, and I've found an ideal way to do that is using finite difference methods. We can also validate theoretical models by doing curve fitting and data analysis. Thermodynamics can be introduced into the introductory physics course by talking about chemical potential of oxygen in blood and air. Osmosis is actually driven by the chemical potential of water. We can talk about entropy and the distribution of states. The Boltzmann factor is really important for the energetics of molecular transitions and chemical processes. Phase transitions are another example related to evaporative cooling, which we've already talked about, and ligand binding, which we also talked about. Random walks and molecular diffusion are a topic that's worth coming back to after students have a little bit more mathematical sophistication. And then lastly, the whole of the electrical introduction can actually be done by relating it to biological systems, in particular ion channels and the membrane voltage that's caused because the membrane, cell membrane acts as a capacitor. And then you bring in standard concepts of electrical things about current, conductance, and resistance, and batteries and RC circuits. And we can finally get to the action potential of the nervous system. So the pedagogical approach that I chose to use is based on guided inquiry, and I've developed self-study guides that are self-contained and are actually quite good for online teaching. Students get activities that keep them busy and engage them with the materials. There's an emphasis on developing skills. A primary one at the beginning is reading and interpreting graphs, learning about semi-log graphs, fitting models to data, using linear regression and non-linear least squares fits, doing residual analysis, again graphical, and an important topic for all of traditional physics is getting students to understand how to model things that change with time. And lastly, the approach to thermodynamics that I use is to use kinetics to motivate thermodynamic concepts. Okay, so here's the cover of the draft book. You'll notice that on the cover there's a diagram. It's called an FD diagram, and we'll be talking a lot about those. And it also has a schematic diagram of the marble game. Here's a picture of the marble game, which is a game that's run by a dice, and the marbles can randomly jump depending on the roll of the dice from one side to the other side, and that simulates diffusion. The marble game jumps simulate Brownian motion of particles in solution. Chapter 1 has a link to a GlowScript program that shows how this works for a collection of 50 randomly moving molecules in a box, and once you start the simulation you can see them all jiggling around, and over a period of time they can move from one box to the other, and the simulation actually keeps track of the number of molecules in each box in the simulation in orange and the FD method in green, which we'll get to soon. Chapter 1 also starts with an introduction to Microsoft Excel, and it starts right from the very beginning with how formulas work. So here's an example of the spreadsheet that students prepare. It has headings in row 1, and column 1 is just a count of turn, which is done by using the formula equals the cell above plus 1. Column B is for the random number, and it's calculated using the formula ran between open paren 1, 200, close paren. And the number in box 1 starts out as 0 and then is calculated using this formula, which is equals, and then the function if the random number is less than or equal to the number in box 1, then make it the number minus 1, comma, the number plus 1. And then the rest of the spreadsheet can be filled in by simply highlighting row 1 and then left double clicking on the blob. And you'll see that the spreadsheet that's made is live. It generates another random instance of the program simply by hitting delete in a blank cell. Chapter 2 introduces students to algorithms, which have a number of features: parameters, variables, an initialization step, and then the main algorithm. I also am really keen on getting students to do unit checks, because in biophysics and physiological modeling, the units don't always match standard SI units. Chapter 3 introduces students to finite difference methods, and an important part of that are the finite difference diagrams, which indicate the rates at which marbles jump from box 1 to box 2 and from box 2 to box 1. Those rates can then be used to derive a finite difference equation. The minus indicates leaving box 1 and the plus indicates going into box 1, which can be rearranged to give a simple FD equation for the small change in the number in box 1 during a small time increment delta t, the time step. Ultimately, students complete a spreadsheet which compares the kinetic Monte Carlo simulation of the marble game with the FD method. This is again is a live spreadsheet, and when you hit delete in a blank cell, it updates, and you can change the number in the box. So I can go from 50 to 500, and students can see how larger systems behave in a more predictable way. In Chapter 4, students modify the marble game to model drug elimination. And the only difference in the model is that now jumps back from box 2 to box 1 are not allowed. The FD diagram for drug elimination is even simpler than the one for the marble game because there's only one direction that marbles can jump. In the chapter, students discover that the model predicts a simple exponential decay in the concentration of the drug as a function of time, as indicated by a linear fit to a semi-log graph. Students also discover that radioactive decay also exhibits a similar exponential decay with time. Chapter 13 outlines how the finite difference approach that students have learned can be applied to standard Newtonian physics, using a problem-based approach to modeling the acceleration of the world's fastest car. Equation 13.1 shows how a small change in position can be predicted knowing the velocity for a small time step. Similarly, a small change in velocity can be predicted using the acceleration and a small time step, resulting in Euler's method. Those two equations can be rearranged to give finite difference definitions of velocity and acceleration. Euler's method can be improved upon using the midpoint algorithm. And using the midpoint algorithm, students are able to discover the kinematics equations. Now that students know what acceleration is, they can then learn how Newton's second law can be used to predict the acceleration if we know the net force. Using the finite difference approach, students are able to show that the work done is equal to the incremental kinetic energy change. By visualizing the small change in velocity in a graph, students are able to see that the area of a small rectangle in this diagram is given by delta V, which is the width of the rectangle, times its height, which is V. So that the area of the small rectangle is just V times delta V. Using that representation, students are able to see that the area is given by the sum, which for students with calculus can be converted into an integral, which evaluates as this, which can then show that the small change in kinetic energy is actually given by the difference in these two expressions, which derives the standard equation for kinetic energy. Ultimately, students can model the performance of the Bugatti Veyron by including its 7-speed transmission and air resistance to show that the simple model can predict the actual performance of the car as a function of time. Chapter 5 introduces a novel model of osmosis, which treats osmosis as the diffusion of water across the cell membrane through an aquaporin protein. Red blood cells are surrounded by flimsy cell membranes that cannot maintain any significant pressure difference between the inside and outside of the cell. but plant cells have cellulose cell walls that can provide a significant pressure difference. To be able to understand how osmosis occurs in that situation, students are introduced to the marble game with gravity. And the way that that's accounted for is by raising the height of one of the boxes, and what happens is that the jump rate in one direction is reduced. For osmosis, that energy difference is provided by PV work, and the energy factor epsilon is given by this expression. As a result, students can formulate a model of diffusion which can account for pressure differences between a rigid plant cell and a bathing solution that it finds itself in. Think of a stalk of celery in a glass of tap water. And that model ends up motivating the Van 't Hoff equation, which defines an osmotic pressure difference in terms of the osmolarity difference between the two boxes. Chapter 6 applies the same approach to one of the most fundamental processes in biochemistry, which is the binding of a ligand to a protein. The example that we use is oxygen binding to myoglobin. Using the finite difference method, students are able to derive an equation for the occupancy of the myoglobin molecule as a function of concentration of oxygen molecules. Students are then introduced to the method of least squares fits to fit the nonlinear equation, and Q is the quality of fit for the sum of the squares of the residuals. Students then use a pre-formatted spreadsheet. By using Solver, students are able to find the best fit value of the binding constant to make the data fit the curve using a properly non-biased least squares fit. In Chapter 7, students model diffusion, starting out with a 3-box model using finite difference methods. By extending the model to more boxes, students are then able to model transient diffusion problems that can't be solved using analytical methods. And those models successfully predict what happens in the GlowScript marble game simulation from the initial conditions and equilibrium when the marbles are evenly distributed between the two boxes. In Chapter 8, students investigate the distribution of marbles between the two boxes in the marble game, and ultimately are able to develop a live spreadsheet that shows the distribution of marbles using a probability density as a function of the fraction of numbers in box 1. This spreadsheet is live, and it shows different distributions generated randomly from the marble game at equilibrium. And by changing the number of marbles, students can see how the distribution changes with the size of the system. In Chapter 9, students stack boxes of air one on top of the other to provide a finite difference model of the isothermal atmosphere. Using that model, they're able to predict the Boltzmann distribution, which then leads to the concept of a Boltzmann factor, which can be used to model a wide variety of molecular systems, starting with liquid-vapor equilibrium. And the model successfully predicts the equilibrium water-vapor pressure as a function of temperature. The same concept can also be applied to ligand binding, resulting in a surprisingly good fit to experimental data for myoglobin occupancy as a function of partial pressure at 5 different temperatures. In Chapter 10, students model random walks in Excel, and they can see various random walks and how they relate to transport, and the standard model of random walks as a spreading Gaussian. In Chapter 11, students model permeation of potassium ions through potassium channels in the membranes of all cells. The permeation of ions through the selectivity filter of the ion channel occurs through a knock-on mechanism. Because there are more potassium ions on the inside of the cell, they tend to diffuse out of the cell, but then get attracted back towards the negative counterions that they left behind. And hence, the cell membrane acts like a capacitor. Using that model, students are able to develop a simplified model of the action potential. Using those concepts, students are able to be introduced to traditional circuit concepts and develop an equivalent circuit model of an ion channel in a cell membrane. Chapter 12 is about applying the same finite difference techniques to modeling the epidemiology of COVID-19, and that will be the subject of a poster. As a teaser, here's a graph showing how a simple SIR model can successfully model the course of the pandemic in America up to today, December 13th, and showing potential outcomes depending on the size of the model population. So in summary, we've talked about physics that life science students want and need. We've talked about how you can start with life science topics from the first class, how finite difference methods can be used instead of or leading to calculus, how scientific modeling and data analysis can be incorporated in the introductory physics class, how we can use kinetics to get to thermodynamics, and how a guided inquiry environment can be used so that students can discover for themselves that science is an evidence-based endeavor with testable hypotheses that are supported by experiment. And just to finish up, the project webpage is at circle4.com/biophysics, and you should look for the links to the chapters and other materials at the top of the page.